So based on my calculation, I got 900,000 cells per mil. If we divide 450,000 to 25 by 25, it means okay, so today I would like to detach my primary ADSC cells from 60 millimeter petri dish into two T25 flasks. So I have two 60 millimeter petri dish. So I will subculture the cells into four T25 flasks. Okay, so before I do the Trypsin initiation, I prepare the media, the complete media, and then I put 3 milliliter of complete media into each T25 flask. Okay, then I will put it all the flask into the incubator just to make it bond. So for detaching the cells or trypsinize the cells, I took the old media and put it into another tube, which is 15 ml of tube, and then I take 3 ml of PBS, the same amount of the previous media, and then I put the PBS into the plate culture to wash the cells. So I'm going to take out the PBS and then add 3 ml of TE 0.05% and incubate 5 minutes inside 0.5% CO2 incubator at 37 degrees for 5 minutes, and then after that detach the cells, put it into the old meat, and then I'm going to centrifuge the cells to make the pellets go to the bottom of the tube and then discard the old media, and then I'm going to count the cells. So after 5 minutes we evaluate if the cell is already detached, then we transfer the cells into the old media to deactivate the trypsin. Make sure there is no cells left or only maybe few cells. It's okay, but I think it's already all the cells already detached and put inside the old media, and then spin the cells. Make sure the balance already balanced. So for these stem cells, I use 1500 RPM or basically 400 G, and I spin it for 5 minutes. Okay, so start from now. Okay, so to count the cells, I put 20 microliter of 1 microliter cell suspension that I've already spin into Appendorf tube, and then I add the same amount of Trypsin blue. So this tube contains 40 microliter 1 by 1 Trypsin blue and cell suspensions, and then I'm going to take 20 microliter from this tube and put it into the counting chamber, and then you calculate the cells in these 4 by 4 big boxes. From here I have around 49 cells, and then I've already counted there was 48 and 45. So basically you have to change into another 4 by 4 boxes. Yes, like this one. And then you make the average of those number of cells. So based on my calculation, I got 900,000 cells per mil. So then I divided by 2, which is 450,000 per T25 flash. So if we divide 450,000 to 25 by 25, it means the cell density is 18,000 cells per centimeter square. So I'm going to, my plan is to seed the cells at 20,000, but it's okay, 15,000 is okay, because even 10,000 cells per centimeter square is enough, but I don't want to push the cells to divide more often, because I'm going to keep one of the pile of them into the into the liquid nitrogen. After 24 hours, we can evaluate the cells on the infrared microscope, and the cells looks fine. It means the subculturing process was successful.